Thank you very much for the kind introduction, and thank you for inviting me. And the answer to your question, to the first question, now I think that people think that CLC5 is expressed in glomeruli as well. When we published this paper, we thought this is secondary to, but now people think that there is a good reason for that. So I was asked to talk about novel therapeutic approaches for primary hyperoxaluria type 1, or PH1 in short. The primary hyperoxalurias are rare autosomal recessive disorders of endogenous oxalate synthesis, and they represent perturbations in glyoxylate metabolism, and glyoxylate is the precursor of oxalate. Oxalate is end product in vertebrates and in mammals, of course, and calcium oxalate crystals are insoluble in body fluids, and they tend to precipitate as kidney stones, as in nephrocalcinosis. So this is a cartoon of a liver cell. This is the cytosol, this is the peroxisome, this is the mitochondria. This is the bad guy, the oxalate, which is the product of oxidation of glyoxylate by lactate dehydrogenase, LDH. And the question is, where do we get glyoxylate from? So basically there are two sources. The first one is glycolate from vegetarian food, which enters the peroxisome. It is oxidized to glyoxylate by GO, glycolate oxidase. And glyoxylate undergoes detoxification in transaminations catalyzed by AGT, alanine glyoxylate aminotransferase, with pyridoxal phosphate as a cofactor. If AGT is missing or deficient, then we get primary hyperoxaluria type 1. There is a buildup of glyoxylate which diffuses into the cytosol, and it is oxidized to oxalate. The second pathway is the catabolism of hydroxyproline, which takes place within the mitochondria. Hydroxyproline is derived from collagen turnover or animal protein. And again, we end up with glyoxylate, which is reduced to glycolate, and glycolate would enter the peroxisome. And this reaction is catalyzed by GRHPR, and which is causing type 2 if it is deficient, and HOGA1 is actually responsible for type 3. So PH1 results from absence, deficiency, or mistargeting of the liver peroxisomal enzyme AGT. We're not sure about the prevalence of PH1. The figure which is often quoted in the literature is 1 to 3 per million in Europe, and I made just a very rough estimate of the prevalence in the central and southern part of the West Bank, and I reached a figure which is anywhere between 10 to 30-fold above what is found in Europe. If you look at pediatric end-stage renal disease units in Europe, in the US, or in Japan, about 1% will be secondary to PH1, whereas in North Africa and the Middle East, and also in Shaare Zedek, 10 to 13% of all pediatric patients with end-stage renal disease has PH1. The phenotype varies tremendously from end-stage kidney disease in infancy to occasional stone formation in adulthood. And if you look at families, you will find wide intrafamilial clinical variability. As I said, initially we have precipitation of calcium oxalates in the kidney, forming kidney stones, nephrocalcinosis, with a gradual decline in kidney function. And once a patient has reached end-stage kidney disease, they develop a devastating multi-system disease known as systemic oxalosis, with calcium oxalate crystals everywhere, in every tissue you can think of: bone, eye, blood vessels, heart, thyroid, and skin. Just a few examples. This is a KUB. It's not an IVP. You can see calcified kidneys. You can see calcium oxalate crystals in the retina. You can see the inability to walk because of fractures and bone deformities. In end-stage oxalosis, the bone marrow is packed with calcium oxalate crystals, and as a result, they develop hepatosplenomegaly due to extramedullary erythropoiesis, and you can see the calcium oxalate penetrating through the skin as well. AGT is encoded by AGXT, and currently there are at least 199 mutations, 3/4 of which are point mutations. About 4 are minor deletions or insertions. Unfortunately, currently there is no approved treatment for PH1, and what we try to do, we try to preserve kidney function by decreasing oxalate production by administering vitamin B6. Vitamin B6 is metabolized to pyridoxal phosphate, and you remember that pyridoxal phosphate is a cofactor of AGT. Beyond the prosthetic effect of vitamin B6, it may work as a chemical chaperone as well. In addition, we try to decrease crystallization by high fluid intake, by citrate, which is an inhibitor of calcium forming salts. And once a patient is end-stage kidney disease, we try to increase oxalate removal by intensive dialysis, which is performed 6 times a week. But we do know that no dialytic modality can cope with the amount of oxalate which is produced by the liver. The ultimate goal would be a combined liver and kidney transplantation, liver as a gene therapy and kidney as replacing the failing organ. So what are the novel approaches? I'm going to touch on 4 different projects, and I'll start with oxalate degrading enzyme, about gene therapy, trying to correct AGT mistargeting, and substrate reduction, which is the most advanced project now. Oxalobacter formigenes is an anaerobic bacterial constituent of the human gut. It degrades oxalate, and it uses it as the sole carbon source energy. And it was expected that oral administration of a lyophilized preparation would degrade intestinal oxalate, would decrease the concentration of oxalate within the gut, and will enhance the flux of oxalate from blood into the gut. But thus far, this has not been proven beneficial in humans. What about gene therapy? PH1 is well defined. We understand the biochemical pathway. So you would say, why don't we try gene therapy, enzyme replacement therapy? I think the main hurdle or the main obstacle is, number one, we need to reach the liver and the peroxisome within the liver. Number 2 is the fact that AGT is actually catalyzing a detoxification reaction. So in fact, you need to repopulate most of the liver in order to get results. So you need high doses of the vector are required for hepatocyte transduction. And we use, or people use, adenoviral mediated vector, which carry its own obstacles. And the obstacles are, it may elicit immune reaction, either to the viral capsid or to the transduced hepatocytes. And we know that vector genomic integration may induce malignancy. Recently, this paper was published showing that you may get some short-term effect of gene therapy, and they use a liver-directed gene therapy system. They use helper-dependent adenoviral vectors encoding AGT under control of a liver-specific promoter, and a single IV injection was given to AGT-deficient mice, where they show that there was some reduction of urinary oxalate. So these are the weeks post-injection, and this is the amount of oxalate, and you can see that this is the negative control. Those were treated with saline, and those were treated with increasing doses of viral particles, and you can see that you have a decrease in the amount of urinary oxalate. When they further analyzed the data, they showed, this is a Western blot, you can see this is, again, saline, this is increasing doses of the vector, and you can see that there is a lot of AGT, which is formed with a high dose of viral particles. This study looked actually at the activity of AGT, and you can see that there is an increase in activity with increasing doses to supraphysiological activity levels as compared to the wild type. And again, when you look at the intracellular compartment, you can see that most of the AGT is in the peroxisome, not in the mitochondria, in the nucleus, and this is all cell lysate. So the question is, I mean, is this good enough? And I think that at the end of the day, the most important message is that what we're trying to get, we try to get as many cells possible that express wild-type AGT rather than the amount of enzymatic activity. Because even if we are able to populate a third of the liver, 2/3 of the liver will still produce huge amounts of oxalates, and this is not good enough. And this impacts on our common practice not to perform auxiliary liver transplant, but rather to remove the entire liver, which is completely normal, and to transplant a lobe or a liver. The next topic would be mitochondrial mistargeting. About 10 to 20% of all Caucasians carry the minor allele haplotype, which includes a polymorphism in position 11, where proline is substituted to leucine, and this generates a cryptic mitochondrial targeting sequence. This on itself does not cause much harm, but in combination with the GLY70R mutation, which is quite common in Europe, unfortunately not in our region, there is actually a phenotype, and it delays folding and dimerization of the AGT molecules. So we do have a misfolded protein with mitochondrial targeting sequence, peroxisomal targeting sequence, and naturally it will be very difficult for a misfolded protein to enter the peroxisome, and that's why it is targeted into the mitochondria. But it retains the catalytic activity. And the only reason that we have PH1 phenotype is because AGT is missing within the peroxisome. A couple of years ago, a group from California performed a chemical genetic screen which yielded molecules that inhibit mitochondrial import. In other words, the gate to the mitochondria was closed, and one agent is DECA, which is a topical antiseptic, and it inhibited translocation of the mutant into the mitochondria and restored peroxisomal localization. They also show some decrease in oxalate accumulation, as I'll show you momentarily. So here you have CHO cells transfected with either the wild type or the mutant. This is staining with AGT. This is a peroxisomal staining, and this is merging. So you can see co-localization. In other words, the AGT is within the peroxisome. Here is the mutant treated with a vehicle, and you see very little of co-localization. And after treatment with DECA, you see that, again, you see more and more of those yellow dots, meaning that, they were able to reroute some of the molecules into the peroxisome. When they looked at the amount of oxalate secreted into the medium, this is the relative oxalate. These are the wild type, these are the mutants. Those were treated with vehicle, they have a lot of oxalate. There was some decrease with DECA, similar decrease with pyridoxal, and the combination had some additive effect. We came across this We came across this paper, which came from Michael Sherman's lab in BU, and he actually suggested a novel approach to recovery of function of mutant proteins by slowing down translation, and use for slowing down translation emetine. So what's the idea? You take emetine, which is known as a translation elongation inhibitor, and you slow down the translation. It will reduce the protein expression somewhat, but will improve its function. And the model which he used was CFTR, which is affected in CF. Emetine is prepared from ipecac roots and has been used years ago for treatment of amoebiasis. Obviously, with metronidazole, we don't need emetine, especially the side effects of emetine are nausea and vomiting. But another virtue of emetine is the fact that it is found in the liver up to a month after a single dose. So we wanted to see whether this can actually affect, with emetine will have an effect for mistargeted AGT in PH1. And the main dilemma that we had initially was how are we going to quantify the rerouting of AGT into the peroxisome? So option number one was to see whether we can do co-staining, but co-staining is quite problematic because it's difficult to subject to statistical analysis. So we say, well, maybe we can separate the intracellular compartment and see whether we can separate the peroxisome from the cytosol in the mitochondria. And it was very time consuming, and at the end of the day, there was very high background. So we decided to do something else and try to develop a method which is based on split GFP. GFP is a green fluorescent protein, and what you do is just split the molecule into two fragments. One of them is hooked to peroxisomal targeting sequence, the other one to AGT, and they have the ability to associate with each other if they are found within the same compartment. So you can see here, if they are in different compartments, no fluorescence, and if they are in the same compartment, then they glow. So we called it glow AGT. So we used CHO cells that were transfected with either wild type or mutant, and we subjected them to emetine treatment. And what we found here, this is actually the corollary of this one. LRM is the mutant, wild type is the normal fluorescence. And you can see that if you subject those cells to emetine, you get an increase in the subpopulations of AGT cells, AGT molecules which are within the peroxisome. We try other translation elongation inhibitors such as GC7. We tried mitochondrial transport inhibitors like the DECA, and we found that actually with another translation inhibitors we get very similar result. We get some additive results if we add a mitochondrial transport inhibitor, and we were very happy about that. But the question is how relevant it is to PH1. I mean, we're talking about transfected CHO cells, and we need to test those on hepatocytes. So luckily, we had access to upside hepatocytes, which are now commercially available. And we get hepatocytes which are derived from liver transplants, either wild type or patients with PH1. And this is actually the same, where are we? Here. This is actually the same mutant that we worked with in the CHO cells, and you can see this is the wild type. You can see that the untreated hepatocyte had very high oxalate, and we have some substantial decrease in oxalate levels in the media with emetin. I just want to conclude this part of the talk by acknowledging the outstanding work of people in our lab, which is directed and under the mentorship of Ruth Belostotsky. People from our division are collaborating and funding agencies. The most developed project is actually the substrate reduction by siRNA. RNA interference is a naturally occurring cellular mechanism for regulating gene expression mediated by small interfering RNA. And we know that synthetic siRNAs can be designed to target endogenous mRNA transcripts of a given gene. And now there are at least 2 pharmaceutical companies I'm aware of, Dicerna and Alnylam, that developed a very efficient delivery system so they can actually send those siRNA inside the liver. So what are the targets? Those companies are trying to target GO. GO is the enzyme which catalyzes glycolate to glyoxylate. And in PH1, there is buildup of glyoxylate because this enzyme is missing. And the theory is that if this is good enough, then we're going to get very little of the substrate, which is converted to oxalate. On the other hand, the readout will be that there's going to be an increased levels of serum glycolate and urinary glycolate. The other target, which is still in preclinical studies by a group from Wake Forest, is to try and target the first enzyme in hydroxyproline metabolism. So I'm going to share with you some of the information that has been published from both companies. A stands for Abigail, but also for Alnylam. And D stands for Judah, but also for Dicerna. So here are studies in healthy rodents. Those are mice and those are rats. And here you have increasing doses of the inhibitor. And what you can see that there is a marked decrease in the amount of mRNA. And with increasing dose of the inhibitor, you get very high levels of serum glycolate. This is true for mice. This is true also for rats. If you take AGT deficient mice, knockout for AGT, and you treat them with the inhibitor, you get a decrease in the amount of urinary oxalate, which mirrors the increase in plasma glycolate. And the fact that, those 2 changes mirrors each other actually point to the fact that glycolate increases as a result of GO inhibition in the liver. The next study was to try to take, again, knockout mice and to feed them either water containing ethylene glycol, which is glycolate, and treat them with either PBS or with the inhibitor. So what you can see here, this is the negative control. Those are knockout mice fed with just water and PBS. This is the positive control, knockout mice with ethylene glycol. And here you have a group which was treated with ethylene glycol and the inhibitor, which is closer to the negative control. And this group is quite interesting because initially they were treated with PBS, and then they introduced the inhibitor, so it went down. Next was to use non-human primates, and again you see the same trend. There is a decrease in mRNA after 15, 29, 155 days, and you have an increase in the amount of glycolate. Thus far, both companies completed phase 1, and Alnylam is recruiting patients for phase 2. So we changed the phenotype, and instead of having patients with hyperoxaluria, we're going to have patients with glycolic aciduria. And the question is this safe enough? So we have two lines of evidence that may suggest that this is safe. The first one is a knockout mice, which was provided by Eduardo Salido. And he's shown that these mice actually have glycolic aciduria with otherwise normal phenotype. We described the first and thus far the only patient with homozygous loss of function mutations in AGO and encoding GO. And it seems that it causes isolated asymptomatic glycolic aciduria. Does this answer all the questions? Obviously, no. We're talking of mice and men. And we do know that oxalate metabolism is not identical in mice and in men. Some people claim that GO may be expressed in the kidney as well. And if this is the case, and you subject the kidneys to very high levels of glycolic acid, will this make any change? Some other people couldn't actually show in the Western blot that GO is found within the kidney. And the most important clinical question is partial reduction in urinary oxalate enough to prevent crystal formation and CKD? I'm not sure that any one of those will turn out to be the treatment for all our patients. I'd rather propose to you the bouquet of flowers model. In other words, we'll have to pick 1, 2, or 3 flowers for each one of our patients on an individual basis, and hopefully the combination will be a cure for this devastating disease. Meanwhile, thank you so much for your attention.
There's room for only one short question. Yes, ma'am.
Thank you, Yaacov, for outstanding lecture as usual. There are some people, you know, they have the tendency to take vitamin C as a complementary. So, do you see more oxalate nephropathy due to vitamin C ingestion, and maybe patients have polymorphisms that make them metabolize the vitamin C differently?
I mean, the question the answer to number one is we don't see a lot of oxalate nephropathy, maybe because we're just overwhelmed with PH1 patients, so we don't really see those. And the second question, whether there are polymorphisms which are associated, I'm not sure. I don't think this has been ever tested, but it's an idea.